This is a sea slug. Isn't it cute? Don't you want to know what's going on inside that little head? Ah, so that's what the sea slug is thinking about. Today, I'll be presenting about reproduction in the sea slug Bergia stephaniae, and how the hormone conopressin and its receptor are expressed in the sea slug's brain. My name is Meagan Olson, and this work is done in collaboration with my mentors, Dr. Cheyenne Tate and Dr. Paul Katz at the University of Massachusetts Amherst. This is the sea slug our lab studies. Its name is Bergia stephaniae. It is classified as a mollusk, gastropod, and a nudibranch. Nudibranch is the taxonomic term for sea slug. Bergia, like many other sea slugs, are simultaneous hermaphrodites. This means that every Bergia has both the male and female sexual systems and can mate with any other Bergia. Reproduction consists of mating and egg-laying behavior, often the same day but not at the same time. The image on the left shows two Bergia mating. The image on the right shows a Bergia laying an egg spiral. So this leads us to our first question. How do nudibranchs coordinate the switch between mating and egg-laying? The hormone conopressin is associated with mating behavior, particularly the male mating system, while egg-laying hormone is associated with egg-laying, which is typically a female behavior. These are images of Bergia's brain. The left image shows conopressin expression in the brain. The bright red spots indicate cells expressing conopressin. The image on the right shows expression of egg-laying hormone in the brain. The bright yellow spots indicate cells expressing egg-laying hormone. By comparing the expression of conopressin and egg-laying hormone in the brain, we can see that the patterns of expression are different between male and female biased hormones. Neuron clusters located on the right side of the brain may have reproductive function, since the reproductive organs are located on the right side of the body. The white rectangle indicates our clusters of interest for conopressin, while the white circle indicates our clusters of interest for egg-laying hormone. This leads us to our next question. Could hormone receptors be responsible for modulating the interactions between these cell clusters, thus coordinating between mating and egg-laying? To investigate this question, we sought to identify the DNA sequence of Bergia's conopressin receptor. Conopressin receptors belong to a class of G-protein coupled receptors and bind conopressin. GPCRs are characterized by having 7 well-conserved transmembrane domains. This allows us to find receptors by comparing their sequences between related taxa. Shown here are the conopressin receptor sequences of 2 related species, Limnaea stagnalis, which is a pond snail, and Theba pisana, which is a garden snail. We use these sequences, along with others, to find a partial sequence for Bergia's conopressin receptor. The transmembrane domains are indicated in blue, but the sequence we found for Bergia only has 4 transmembrane domains, suggesting that it is a partial sequence. We predict that the full sequence, shown by the dotted box, contains the last 3 transmembrane domains. Based off the partial sequence we found, we designed probes specific for Bergia's conopressin receptor gene. The probes contain short pieces of DNA that are complementary to the mRNA of the conopressin receptor, causing them to bind. Once the probes are bound to the conopressin receptor mRNA, we add fluorescent markers which bind to the DNA probes. This describes in situ hybridization reaction, which is a technique that allows us to visualize the mRNA of the conopressin receptor using fluorescence. This is what the end result looks like. The cyan fluorescence represents the mRNA of the conopressin receptor. This shows us that the conopressin receptor is broadly expressed across Bergia's brain. This shows the conopressin hormone in red and the conopressin receptor in cyan. Both the hormone and the receptor is shown together on the same brain in the middle. These images show how the conopressin receptors often co-localize with the conopressin hormone, including in the asymmetric region indicated by the white rectangle. Arrows point to other regions of co-localization as well. In examining the conopressin receptor in cyan with egg-laying hormone in yellow, we do not see co-localization. However, the conopressin receptor does have an asymmetric cluster of neurons near the egg-laying hormone cluster. This suggests that they may interact through a different mechanism than we originally thought. To conclude, the nudibranch Bergia-Stephaniae is a hermaphrodite. Hormones play vital roles in coordinating male and female behaviors. We can localize hormones in the brain using in situ hybridization, but this does not show interactions between neurons. We thought neurons expressing female egg-laying hormone may receive input from male hormone-expressing neurons via conopressin receptors. However, we found that conopressin receptors do not co-localize in asymmetric clusters of putative reproductive neurons expressing egg-laying hormone. We did find that conopressin receptor neurons are near the egg-laying hormone cluster, with proximity perhaps indicating a different mechanism for interaction. And finally, we found that conopressin receptors often co-localize with conopressin, indicating there may be some feedback interaction between the hormone and its receptor. All of that inside that cute little head. I would like to acknowledge everyone in the CATS lab for supporting me in my research, as well as Jim and Maya at the light microscopy facility, who were instrumental in helping us image the brain. This work was funded by UMass Amherst as well as the NIH. If you would like to learn more about our research, check out our lab by using the link below. Thank you!